All right, good morning, everyone. Welcome to week 13 of our reviews. We're nearly to the end of the semester, and actually last week is going to be our last review. So before we start jumping into the content, I want to briefly go over what you guys can expect from the next few weeks. So from what I understand, your professors should have just finished the lectures on the Tca cycle, and you guys are about to start the electron transport chain. So I think that these two chapters are very, very content heavy, whereas some of our other units like amino acids, like enzyme kinetics, like those kinds of topics are a lot easier to do practice problems for. I think the toughest part is that they're just confusing and there's a lot of content. And so for the next few weeks, I'm going to shift over to mostly kind of leaning more toward your professor's PowerPoints than practice problems, because I think that that's an easier way to learn this material. And we're going to try to go into depth and explain a couple of things that I think can be difficult to understand when they're not put into perspective. So yeah, the other thing is I know the electron transport chain is 3 lectures when you'd normally have only 2, so this Thursday and next Tuesday. So because of that, next Wednesday's review, I'm going to do my best to try to compact it into that hour and 15 minutes. I'm going to do my best to cover the entire electron transport chain, but I may very well only have time to just very briefly go over the major points. Okay, so without wasting any more time, I'm just going to go ahead and jump right in. So in the past few weeks, we've been talking about glycolysis, gluconeogenesis, and glycogen, and all these other things. Okay, but after glucose is converted into 2 pyruvate molecules, after your NADH is made, we've got a couple of options, and we've talked about these options very briefly before. In yeast and in animal cells, you can do some form of anaerobic respiration to get to either alcohol in yeast or lactate for us. Okay, and then the points of that are two things. First, it recycles our NADH. We need NAD plus in order to do more glycolysis, and so we can recycle NADH into NAD plus and use that for more reactions. Or, and this is the more ideal situation, in the presence of oxygen, we can go through what, like it says here, the tricarboxylic acid, the citric acid cycle, the Krebs cycle. All 3 are used interchangeably. And this gives us a lot more energy, and this gives us a lot more reduction potential to make ATP, right? So kind of the basic steps, actually, you guys are flipped. So over here, the basic steps are we start with glycolysis to get to pyruvate. We get a little bit of energy, but not a lot. Then from pyruvate, we get, we basically oxidize completely to CO2. We go through the citric acid cycle, and what we get out of that is a lot more NADH, some FADH2, things like that. And then those molecules go through the electron transport chain, and they make a proton gradient using the hydrogens that they have, and give us a lot of ATP using what's called the proton motive force. And we're going to talk about that in a bit more depth next week, but I just want to very briefly go over. Okay, so reducing equivalents that are made are reoxidized in the electron transport chain, with oxidation being coupled to the synthesis of ATP. Yeah, okay, like we talked about. Respiration is the metabolic process for ATP production in which inorganic molecules function as electron acceptors. Okay. Not a lot there, to be honest. So let's talk about this. So from glycolysis, we make 4 ATP, really a net of 2 ATP and 2 NADH, right? We get another NADH from the conversion of pyruvate to acetyl Coa. We get 3 NADH and 1 FADH2 from the citric acid cycle. And then the electron transport chain right here, further oxidation of pyruvate yields more than 30 ATP per molecule of glucose. This is where we get most of our energy. However, again, it requires oxygen and it's a slower reaction, so we can only do it under certain conditions. Okay, so pyruvate, like we talked about, gosh, several weeks ago at this point, is considered the most connected metabolite. It's connected to the most number of things of anything on that metabolic map that you saw with a million different pathways. Okay, so when it goes into the mitochondria, as it says here, all in eukaryotes, all these occur in the mitochondria. So first, it's transported in. Okay, then it'll get converted into acetyl CoA. It'll get decarboxylated. Okay, so this decarboxylation releases a CO2 and forms an NADH. Okay, so if we keep that running tally, we've got a net of 2 ATP, and we got 2 from glycolysis. We just got another 2, so 4 NADH. And where did we get 4 from? Remember that we have 2 pyruvate per 1 of glucose. So there's only one NADH reaction in ATP. It's the glyceraldehyde 3-phosphate dehydrogenase reaction. Okay, but there are 2 of them since each are 3 carbon molecules, and that's something that's really important. We talk a lot about the names of these molecules: glucose, fructose, glyceraldehyde, all these things. I'm going to start shifting a little bit more toward how many carbons does each molecule have, because I think that that's a really important way to keep track of the tricarboxylic acid cycle. So pyruvate is 3 carbons. We lose 1 from CO2, and we're left with a 2-carbon molecule entering the citric acid cycle. And there's an important thing to note here. Fatty acids enter at the level of acetyl CoA, and it says here through beta oxidation. So this is something that you're going to talk about a lot more in Biochem 2, but it's a really important topic to note that we can get energy from fats. And I think that's something you also learned a few weeks ago when your professors might have talked about the keto diet. Okay? So this is the cycle, and I'd like to very briefly go over it before we go into a ton of depth. So one thing, just before we even start, this is a 2-carbon molecule. Every turn of the cycle releases 2 CO2. Okay, so in other words, our net carbon gain in this cycle is 0. We start with 2, we release 2. Okay, so that's one important thing to note, because this is I want to say carbon neutral, if that makes sense, right? We're not adding extra carbon, and that's why we're not growing. Okay, so your first reaction is acetyl CoA plus water plus oxaloacetate using the enzyme citrate synthase. Okay, and they'll form, so oxaloacetate is a 4-carbon molecule, citrate is 6 carbons, okay? And then a conatase will isomerize it to isocitrate, which isocitrate, kind of like leucine and isoleucine, they're isomers of one another, so this is still 6 carbons. Then isocitrate dehydrogenase, we have a dehydrogenase, so we're forming some kind of oxidized product, and we're releasing CO2. So 6 carbons minus 1 carbon, since we released the CO2, is 5 carbons. Okay. And then, excuse me. So this 5-carbon molecule is alpha ketoglutarate. Alpha ketoglutarate with another dehydrogenase, so we get another NADH, and we release another CO2. 5 carbons minus 1 carbon gives us 4 carbons. Okay, and that gives us, because we're also introducing, there it is, the CoA group, that gives us what's called succinyl CoA. Okay, and succinyl CoA is released in this reaction, in the next reaction. We're still not using or losing any carbons, and it's regenerated basically. It's formed to make this thing, and then it's released right away. Okay, so this is a little bit of a weird reaction. So this reaction, the succinyl reaction, uses an inorganic phosphate and a GDP to make GTP. Okay, in other words, we just got energy out of this. Okay, but because everything that we've been doing has been in terms of ATP thus far, and because nucleoside diphosphate kinase can convert GTP to ATP, yes, technically this is GTP that we get out of it, but they're almost interchangeable for the purposes of this class. They do have different functions later down the line, and that's again a Biochem 2 topic, but the amount of energy that we get is about the same. They both release about the same amount of energy because they both have those really high energy phosphate bonds. Okay, now succinate is what's formed after that, and succinate is once again 4 carbons. We have not changed the number of carbons that we have. Succinate dehydrogenase gives us an FADH2. However, one thing to note here is our other dehydrogenase reactions have used CO2. This one does not, so we're still at 4 carbons. Okay, the fumarate reaction of fumarate to malate with water, still 4 carbons, we haven't lost any. And then malate dehydrogenase, we make another NADH, and we're left with our 4-carbon oxaloacetate. Okay, so I think that doing this in terms of both name and number of carbon can be really effective. Okay, all right, so we're just going to jump right in. So coenzyme A, which acetyl CoA, succinyl CoA both need, is it needs vitamin B5 or pantothenic acid in order to be synthesized. Okay, so let me see if I can zoom out a little bit. This is the CoA group. Okay, and the functionality is mediated by sulfhydryl groups. Okay, all right. So this is kind of putting that together. This is what that's all about. Okay, so you have some nucleophile attack. So this is whatever carbon unit you have to attach the CoA group to it. Okay, I'm sorry, not to attach the CoA group, to release the CoA group. So this is the CoA-SH group that we talk about a lot. So when acetyl-CoA, when the acetyl group is transferred away, coenzyme A activates this substrate. In other words, this can only be formed because this, thinking back to organic chemistry, is a good leaving group. Okay? Leaving group. Okay? So that makes this reaction more favorable rather than trying to separate what would be some kind of acetate molecule with this. This bond would not be very easy to break. And so by adding a coenzyme A to it, we can make it a great leaving group. And by making it a great leaving group, we can make the reaction more favorable. Okay? Just like in gluconeogenesis, we go through weirder or like different reactions from glycolysis to make that reaction more favorable as well. Okay? All right. So there's excuse me. So there's an example of just the energy difference. Cleaving this ethyl acetate is spontaneous at about -20 kilojoules per mole. Cleaving acetyl CoA with coenzyme A in it is -31.5. That's about as much as ATP hydrolysis. So this is definitely a lot more spontaneous and a lot faster. Okay? All right. So thiamine pyrophosphate. We don't really talk about this much. So what's the point of it? So TPP, thiamine pyrophosphate, enzymes catalyze the decarboxylations of alpha keto acids. In alpha keto acid, well, first of all, we have alpha ketoglutarate. But we also have, remember, this is not just a glucose-dependent process. We can have things like fatty acids joining at the level of acetyl-CoA. Thiamine pyrophosphate can help with that. Okay, and that's vitamin B1, by the way. Okay, all right. Yes, so here's a great example. And so you start with your alpha cleavage to get to this point, and then this is pyruvate decarboxylase will give I'm sorry, this is not pyruvate, I believe. But the point is that it'll give us the acetyl group that we can just add enzyme CoA to. Okay. Excuse me. This I think is a bit more background reading than anything, so I'm not going to cover it in a ton of depth. But it is, so lipoic acid, it's involved in electron transfer. I haven't seen these kinds of questions asked, though it couldn't hurt to learn. It will come up next semester. All right, so now we get into the kind of meat and potatoes of this unit, which is the part that's kind of tough. And this is the part where we talk a lot about the actual mechanisms of these reactions. Okay, so I'm going to start off with, this is pyruvate dehydrogenase. So we finish glycolysis, and we're not yet at the Tca cycle. So this is a single reaction, the middle bit, from to acetyl-CoA. So this reaction takes 3 steps. Okay, the first step is the thiamine pyrophosphate reaction. By transferring pyruvate to thiamine pyrophosphate, we almost activate this enzyme, if that makes sense. Okay. So I'm sorry, not enzyme. We almost activate this protein. Just like attaching it to a CoA group can activate it and make it more favorable to react with something, this does something similar. Okay. So now the hydroxyethyl group is transferred to lipoic acid and oxidized to form acetyl dihydrolipoate. I'm not great with the names here. So hydroxyethyl is this group. This is what's left of the pyruvate. Okay. And now if you notice, it's right here, but rather than being a hydroxyethyl, it's got that double bond, okay? So we've oxidized it here, all right? And now finally, acetyl group is transferred to the CoA group, and then to regenerate the lipoic acid, so your lipoic acid is basically a disulfide bond, okay? We oxidize sulfur here and here by giving it something to attach to, and now it's got these two hydrogens. I'm sorry, we reduced sulfur. I always do that backwards. We reduced sulfur, and now we oxidize it. How do we NAD plus to NADH? Okay. And so this is the oxidation reaction, and it has nothing to do with the pyruvate, as an interesting fact. Okay, so this is one reaction. Thankfully, we don't go into a ton of depth with all of these. Yes, and your professors went into a lot of depth on each individual step. Okay, so there is a lot of stoichiometry. There's a lot of subunits. It's very complicated. This is a little bit beyond the scope of this class, though. Okay. This is something that I would encourage you all to know. In eukaryotes, we have a lot more of subunit E2. E1 is a tetramer and has two subunits, and E3 is a dimer. Okay. All right. So this is what it looks like in real life. And so here's a little bit more depth. So the lipoamide. So lost my mouse. There you go. These really long fatty acid structures, they're a swinging arm. Because they've got such a long side, they can swing around between the other subunits, okay? And then the second point on there is it's a flavoenzyme, E3, okay? So FADH2 stands for reduced flavin adenine dinucleotide, okay? So in other words, F is flavin. So when it says it's a flavoenzyme, all it's saying is it's going to be dealing directly with FAD or FADH2. Okay? It's not directly oxidizing or reducing NAD and NADH. The FADH2 does that for us. Okay? So it's a kind of two steps. The first thing gets oxidized, and then that oxidizes the next thing. Or, I'm sorry, reduces. Yes. Okay? So here's how it looks all together. We're going to go ahead and skip past that. Okay. So now we're on to our first reaction. Okay. So citrate synthase. So like we said, it brings oxaloacetate and acetyl CoA and makes them react. So this is a pretty interesting reaction because it's got it's base catalyzed, right? So you have this base abstracts this hydrogen, and then that helps stabilize this oxygen when this carbon attacks it. And when this carbon attacks it, we get this attachment right here. And then, excuse me, this water comes in and kicks that out. Okay? Excuse me. So it's a carbon-carbon condensation reaction, and the product is citrate. So why is this called the Tca cycle? We have a carboxylic acid, another carboxylic acid, and a third carboxylic acid. So it's a tricarboxylic acid. Okay? So that's why it can be called the Tca cycle, the citric acid cycle, the Krebs cycle, because Krebs is the person who found it or discovered it. Okay. Now, an important point here, this reaction is very spontaneous. This reaction is about as spontaneous as the ATP hydrolysis. Okay, that makes it pretty much irreversible under normal conditions. Okay, however, it can be allosterically inhibited by NADH and succinyl CoA. So when we talked about NADH and succinyl CoA, NADH is what we're trying to make from the Tca cycle. So it makes sense, right? We're not going to want to keep making more citrate if the acetyl CoA and the oxaloacetate could be used for other things. Okay? And succinyl CoA is about halfway through the citric acid cycle, which also makes sense. And one important thing to note here is this is a CoA, right? And I know that doesn't make a ton of sense, but what I mean by that is this also uses coenzyme A, both of these do. So let's think about what that tells us. If we have a lot of succinyl CoA, that means that the amount of coenzyme A that we have like floating around freely is pretty low. Okay, So this tells us we could react more of our acetyl CoA to release some, but we don't need it. It's more useful here as acetyl CoA where it can be used for other things, okay? Okay, so one of the questions that I had when I first learned this in my intro bio class is why is the isomerase that changes citrate into isocitrate called aconitase? Doesn't make sense. The answer is actually a little bit easier than you might think. It's because there's an intermediate that we don't show, and it's called aconitate. So aconitase it is overall isomerizing citrate, but what it's doing is it's converting citrate into into aconitate, and then aconitate is converted into isocitrate after that. Okay. So that's using a dehydration and rehydration. Okay. And as for why this is important, it sets us up for future oxidation reactions. So remember that we're going to be losing a lot of CO2s, and that those CO2s are going to be well, actually, I'll show you the CO2s in a bit. I don't want to spoil it. Okay. So this is something that you guys are going to have to get a little bit used to. It's one of the weirder things about this this unit. Really, this and next chapter is what kinds of things are in the enzymes. So in this enzyme, there's a 4-iron, 4-sulfur cluster, okay? And it binds to the side chain using 3 cysteine residues, which each have their own sulfurs. So the iron is very easily lost, and that can deactivate this enzyme. So it almost has a built-in way to regulate itself, right? Okay. So here's the reaction put all together with all of the arrows and the irons and everything like that, okay? So let's talk about inhibitors. So fluoroacetate, rat poison, is incredibly toxic by inhibiting the Tca cycle. So by inhibiting aconitase, and actually I'm going to underline suicide substrate so I don't forget to talk about it. So I'm sorry. So it's a great inhibitor of aconitase. How? Fluoroacetate has a fairly similar structure to pyruvate, and so by going through ATP synthetase, you can get fluoroacetyl CoA, very similar to acetyl CoA. So now this plus oxaloacetate will give us the citrate synthase reaction, which will give us fluorocitrate. Fluorocitrate can bind to aconitase as a suicide substrate. And if you guys remember suicide substrates, they're the ones that are basically irreversible inhibitors. It'll bind and it'll never let go. And if all of our citrate synthase is bound, or I'm sorry, all of our aconitase is bound, then we can no longer go through the entire Tca cycle, and we lose all of that energy. Okay? Right. So the next reaction is the isocitrate dehydrogenase reaction. So the next two reactions are actually both dehydrogenations. And so what is the structure? Which carbon is losing its carboxylic acid? The second one. Okay. So this carboxylic acid, if you notice, is gone. And as a result, so this that was here is gone. And this alcohol group is now oxidized into a some form of a ketone. Okay, and that's actually why this is called a keto acid, right? Ketone plus carboxylic acid. Okay, and so this is one of those keto acids that we talked about. Okay, all right. So now once again, fairly similar reaction. We're gonna have, excuse me, this carboxylic acid group leave as CO2. And we're going to have NAD plus reduced into NADH. I always get that mixed up. And we add coenzyme A to this alpha ketoglutarate. Okay. So like I said, it's fairly similar to the pyruvate dehydrogenase complex, and it's kind of similar to the previous complex, though in more in terms of the broad strokes. And actually, as a fun fact, the pyruvate dehydrogenase complex and the alpha ketoglutarate dehydrogenase complex share their E3 subunits. So the E3 subunit is that flavoenzyme that that reduces FAD plus into, or I'm sorry, FAD into FADH2, and then that will reduce NAD into NADH. Okay. Now we get succinyl CoA. And succinyl CoA is going to give us that GTP that we talked about. So this is a very, very, very spontaneous reaction that releases a ton of free energy. So this free energy that's released drives the phosphorylation of GDP to GTP, and that phosphorylates ADP into ATP. Okay. So one thing that I really want to point out here is this is the only substrate level phosphorylation in the TCA cycle. I've seen this question on several different exams in the past. They love this question because it shows whether you're paying attention. Okay. And as another important note, in plants and bacteria, you don't really have to go through GTP. You can go straight to ATP. Okay. So we've gotten through, let's see, we've just made succinate. So we're to here. Okay. And thus far, let's kind of take stock of what we've done. We've made the NADH from pyruvate dehydrogenation. We've made 2 more NADH from isocitrate and alpha-ketoglutarate dehydrogenation. And we've made 1 ATP. All that's left is FADH2 and 1 more ATP. Or I'm sorry, no, not ATP, 1 more NADH. And we've gotten all the energy out of this cycle, excuse me. So, like we said, we've used the 2-carbon unit and linked it to oxaloacetate to make citric. And we've lost 2 carbons. So we started with the 2 carbons we put in, plus this original 4-carbon thing. We're back down to a 4-carbon thing, and we've lost the 2 carbons that we put in. Okay? All right, so succinate dehydrogenase. So I want to point out here, this is not exergonic, or I should say this is not very exergonic. And so if you've taken an introductory bio course, you might have learned that NADH gives more ATP than FADH2. Well, the reason is because NADH has more reducing power, reducing potential, etc. Okay, than FADH2. And so as a result, because the succinate dehydrogenase reaction isn't very spontaneous, isn't very exergonic, you can't get NADH, so you get FADH2. Okay, and I want to point out why this may not be the most exergonic. We've got a double bond here. We've got a carboxylic acid here. This thing is very polar. And so there's so many polar bonds pulling every carbon in every direction, and it makes it pretty unstable. Okay, and it's associated with the mitochondrial membrane and contains 3 iron sulfur clusters. That's a little bit of background. Okay, so we've been talking about FAD kind of a little bit as just here's something, let's not actually talk about it. This is where we're going to start talking about it a little bit. So riboflavin, or vitamin B2, is required for the synthesis of FMN and FAD. So FMN is kind of the F. It's flavin mononucleotide. And like we talked about earlier, FADH2 is flavin adenine dinucleotide. So the flavin is really the same between them. And that's something that you'll see. Oh no, sorry, this is the, so you'll see FMN, and then FAD is FMN plus some stuff, basically. And so, so these groups are found tightly bound, as tightly bound prosthetic groups in flavoenzymes. So that's why we need riboflavin, okay? So the 3 redox states, they each have 3. So they can participate in 1 electron transfer or 2 electron transfers, okay? So that's what we see here from FAD or FMN in this top corner. You can either do 1 electron transfer to FADH, 2 electron transfer to FADH2, and then you can also go backwards. So yes, so this diagonal is a 1 electron reduction. And then finally, what's happening here is we can lose another hydrogen and give us this semiquinone anion, which is going to come up in the Tca cycle. Okay, all right. So the isooctanoate Isoaloxazine ring, sorry, that always trips me up a little bit, is covalently linked to a histidine in succinate dehydrogenase. Okay, so why is that important? No, okay, sorry, I had to always misremember how the slides go. Okay, so why is it important that it's linked to a histidine? So let's look at that specific carbon that's linked to a histidine and see what's going on with it. Or rather, I should say what's going on with the ring, not just this carbon. Okay, so this ring is linked to a histidine. In other words, when we talked about these are found as sorry, I lost my place. Up here, excuse me. As it being associated with the mitochondrial membrane, it being associated with the mitochondrial membrane, this is how it attaches, because remember that succinate dehydrogenase, this FAD is going to be made right here on the membrane, okay? And then it also contains the 3 iron clusters, and this one is 2 iron, 2 sulfur. This is 3 iron, 4 sulfur, 4 iron, 4 sulfur, okay? And it's bound to the inner membrane of the mitochondria, and the electrons enter the electron transport chain directly, okay? So we're going to talk about succinate dehydrogenase both in the Tca cycle and a little bit later when we talk about next week in the ETC, okay? All right, so iron-sulfur clusters. I've talked about them. I haven't really talked a lot. So they're bound to proteins via cysteine side chains. We talked about that. So 2,2 and 4,4 iron-sulfur clusters contain inorganic sulfur, okay? And they typically cycle between oxidation states differing by 1 unit. In other words, they transfer 1 electron each time in each redox. So we're going to be talking about quite a lot of proteins that have these iron-sulfur clusters and a lot of heme groups, which can also transfer electrons using iron in the next few slides and in the next few weeks, really, okay? So the spacing is optimized to facilitate electron transfer, okay? So multiple redox centers would constitute an electron wire. And that's what we're going to see, once again, next week is going to be, we're going to transfer from this electron cluster to this one, to this one, to this one, to this one, and so on, okay? So before we get into this, I'm going to take a break. Does anyone have any questions? Okay, all right. So iron-sulfur clusters are essential. So for example, in the gene coding for taxin, if that gene is mutated and is unable to work properly, you get what's called Friedreich's ataxia, okay? And it causes damage to the nervous system, speech problems, heart disease, all kinds of things, right? So what does that have to do with the iron-sulfur clusters? Frataxin participates in the assembly and maintenance of these clusters in the mitochondrial enzymes, and most specifically in aconitase, okay? All right, so now, before we get into the this actual reaction, I want to point out what it says down here. Stereospecific hydration and L-malate. So when we've been talking about sugars, we've been talking about glucose, fructose, mannose, all those other things, and we've been talking about them as, for some reason, they're always in their D forms. They're not here. This is in its L form, and this is one of the weird ones that I highly encourage you to know. I have seen it on exam before. But this is a fairly similar reaction, fairly simple reaction. This side gets an OH, this side gets an H, and we're done. Okay. All right. So, Mallet dehydrogenase. So this reaction, this is the last step of the cycle, okay? And it's very endergonic. So that's something that's really important. This reaction doesn't really want to happen. And when we talked about NADH and FADH2 before, it kind of makes sense why. Because NADH is a lot of reducing power. So we would need a lot of power to make it. That's why this reaction isn't very spontaneous. And yet it's okay that it's not spontaneous because it's, as your book says, pulled along by the citrate synthase reaction. In other words, despite malate oxalate not being very spontaneous, the coupled reactions are spontaneous. All right. So, yes. So I'm going to pause here for a moment because this is technically the end. Does anyone have any questions? Okay. All right. Now I'll go ahead and jump in. So before we jump into the specific content, before we jump into the next few slides, I want to point out one thing. We talked about malate and oxaloacetate before, and specifically we talked about them in the gluconeogenesis section. Okay, we talked about how pyruvate in the mitochondria can leave the mitochondria. How? By being, or I should say, how pyruvate in the mitochondria that's converted to oxaloacetate for gluconeogenesis can leave. And that's through malate dehydrogenase. So we talked about these reactions before. When pyruvate is converted to oxaloacetate, it's then converted to malate. Malate can leave the mitochondria and be converted back into oxaloacetate. Okay, so that's something that I think we might be forgetting a little bit, because it's one of those reactions that's kind of glossed over, given that there's so many others in gluconeogenesis, but it is quite important to see that these two cycles connect quite a bit. Okay. All right, so there's the overall reaction, and as said, for each acetyl-CoA entering the cycle, we get 3 NADH, 1 FADH2, and 1 ATP. But remember, that's per acetyl-CoA. And you get 1 acetyl-CoA from 1 pyruvate, and you get 2 pyruvate per 1 glucose. In other words, we have to double this whole thing. All right, and actually, if we put together what happens in glycolysis in the Tca cycle, that's what we get below. So from glycolysis in the Tca cycle, we get 6 carbon dioxide. So glucose is completely converted to CO2. We get 4 ATP, so some energy, 2 FADH2, and 10 NADH. These two are where the energy really comes in, right? We talked about getting more than 30 ATP. We're going to talk about next week. The electron transport chain is how we get to that 30, or that more than 30. Okay. All right. And again, yeah, it makes an additional 34 molecules of ATP by electron transport and oxidative phosphorylation. Okay. So this so far has been all about the catabolic functions, right? We've been talking about we're going to break all this stuff down, but it's got some anabolic functions too. So this is kind of, and I need to zoom out a little bit more. So this is the connection between the citric acid cycle and everything else. So like I mentioned earlier, your oxaloacetate is how you go back to carbohydrate. So this is the path for gluconeogenesis. This has plenty of side products that you can make, including amino acids, which are all over this area, phenylalanine, tyrosine, and tryptophan, or serine, glycine, cysteine, etc. From pyruvate, you can make these 3 amino acids, alanine, leucine, and valine. Then acetyl-CoA can give us fatty acids, it can give us steroids, or it can give us acetoacetyl-CoA, which we'll talk about. I think that's a Biochem 2 topic, actually. Alpha-ketoglutarate as an alpha-keto acid can help us get to glutamate, arginine, proline, glutamine, and then eventually the purine nucleotides. Fumarate and oxaloacetate can give us aspartate, asparagine, threonine, isoleucine, methionine, lysine, and then the pyrimidine nucleotides, and so on and so forth. And this is just a small part of it, but this is just to show how interconnected all of these reactions are, how resourceful our body can be in building up from these intermediates that we get when we break stuff down. Okay, that's something that I personally think is fascinating. Okay, so however, when we do that, we might run out of our intermediates. So for example, if we end up using all of our alpha-ketoglutarate to make glutamate, and then whatever else we make from glutamate afterwards, we no longer have alpha-ketoglutarate to put through this cycle to get energy out. Okay, that's a problem for obvious reasons. And that's why there's these anaplerotic reactions. And these are reactions that I'd strongly encourage you all to have ingrained in your memory, because I've seen one of these on pretty much every single exam that I've seen. Okay, so how do we make more? We talked about this also with gluconeogenesis. You can go from phosphoenolpyruvate to oxaloacetate, and oxaloacetate and malate are near interchangeable. You can go from pyruvate through oxaloacetate and malate. Okay? So this, like it says right here, pyruvate carboxylase also functions in gluconeogenesis, right? And amino acid catabolism, in other words, rather than forming glutamate, going backwards to form alpha ketoglutarate, instead of forming aspartate, going backwards to form fumarate or oxaloacetate, that can give us some Tca cycle intermediates. But one thing that I think is really interesting is CO2 is almost always a waste product. And yet here we're using it as one of these anaplerotic reactions. In other words, we're using it for anabolism. It's not a waste product, it's a reactant that we need. Okay? And so these anaplerotic reactions, again, memorize, know these well. Okay? So pyruvate carboxylase is the most important anaplerotic reaction. It gives us that direct link between glycolysis and Tca cycle. And we've got regulation. So when you've got too much acetyl Coa, really, right? So when acetyl Coa is greater than oxaloacetate, well, let's think about what that means for our body. It means that we've put too much through glycolysis that we can't keep up with it. So what can we do? Rather than convert pyruvate into more acetyl Coa and make the problem worse, we can convert it to oxaloacetate. So now instead of making a ton more acetyl Coa, we make oxaloacetate so that the supply of each is reasonable so that we can have more of the Tca cycle activated. Okay? And again, we have to get into regulation. So this cycle is regulated by these 5 things. Okay? And before we even start looking at the stuff on the left, and actually I want to not even talk about them yet. Okay? Let's take some guesses. NAD plus, NADH. One is going to activate, one is going to inhibit the cycle. Which do you think is which? And we can talk about the specific places afterwards, but just before we even get into that, I think we can pretty easily figure out NAD plus will activate the cycle, NADH will inhibit the cycle. Inhibit the cycle. Same with ADP. ADP tells us we don't have a ton of energy. We're going to go through the Tca cycle to get more. ATP tells us we have enough energy. We don't need the cycle to keep going. And Succinate Coa tells us we've gotten a lot of energy out of this. We might not need any more. So now let's look at this and see. So Pyruvate Dehydrogenase is inhibited by acetyl Coa. And this makes sense because it's pyruvate's other option, pyruvate carboxylase, is activated by acetyl Coa. So you'll either make acetyl Coa or you'll make oxaloacetate, depending on what you need. And acetyl Coa tells us that. It's inhibited by NADH and activated, it's inhibited by NADH and ATP, which again makes sense. And it's activated by NAD+, which all, reasonable. And it's activated by coenzyme A. When we have a ton of coenzyme A, we can use that to make energy, right? Coenzyme A is kind of like that priming step. It helps us figure out, it helps us get ready for energy production. So if we've got a lot of it, we can use it, okay? Citrate synthase is only ever inhibited, and it's inhibited by the 3 things you would expect: NADH, ATP, Succinate Coa, okay? Isocitrate, or I should say isocitrate dehydrogenase. When you regenerate NAD+ by entering lactate, does it somehow relate to the cycle as well? Kind of. So I'm going to kind of draw over here so that I have room. So you've got pyruvate here. So pyruvate, and I'm going to go ahead and skip the yeast one because it's very similar to the pyruvate to a lactate reaction, just it makes alcohol instead. So you've got a choice between with oxygen and without oxygen. When you don't have oxygen, you go to lactate. And the reason you go to lactate is because it gives us NAD+. And that's it. So NAD+, this is helpful for us because glycolysis needs NAD+. If you don't have any, you can't make any of the ATP out of glycolysis. So this literally just regenerates NAD+, and it doesn't give us any more energy. Regenerates NAD+ for glycolysis. Okay. Whereas the Tca cycle makes more NADH for a bigger payout. For a bigger payout of ATP. So it's a matter of what are our conditions? Can we go for this longer cycle that needs oxygen and get a ton of energy out of it? Or do we need this energy right this second? We don't have any oxygen, so we're just going to remake our NAD+ so that we can just make a little bit of ATP quickly. And so that's kind of the debate your body goes through, okay? It's a great question, though. Okay. So isocitrate dehydrogenase. This one is kind of classic. So this one most specifically, and actually the next one is different, but this one specifically deals with energy, right? It's forming, this does not have it. It's forming NADH. So since it's forming NADH for us, it makes sense that it would be inhibited by NADH and ATP and activated by ADP. It's the energy reaction, all right? The alpha-ketoglutarate dehydrogenase reaction, and this is actually the last point of regulation here. So it's activated by AMP, which it's not ADP, but it's fairly similar in terms of the idea. If you have AMP, you don't have a lot of energy. And so the fact that it's activated by AMP is kind of similar logic to the ADP. The fact that it's inhibited by NADH, once again, it's forming NADH, so that makes sense. And it's inhibited by Succinate Coa, which is its direct product, which makes sense, okay? This cycle is really great at regulating itself, okay? And we're going to be going into a lot of depth, low energy status, ATP inhibits those reactions, AMP and ADP activate, and yes. So why does Succinate Coa inhibit? It tells us that the cycle is saturated. So in the same way that if you've got a high amount of acetyl CoA, it tells you don't have enough oxaloacetate. Well, now Succinate Coa is kind of serving a similar purpose. We've got so much Succinate Coa that we can't keep going through these reactions, and so it tells your body, stop for a moment, let us catch up, and then you can react, okay? All right, so once again, we are returning to pyruvate dehydrogenase. We very briefly talked about the idea that it's inhibited by acetyl CoA and NADH, but there's a little bit more depth in that. And specifically, we're going to talk about serine phosphorylation. So the E1 enzyme from the PDH complex. So this is the one that involves thiamine pyrophosphate. When you phosphorylate this serine on that enzyme, it inactivates the E1 enzyme, and thus it pauses everything, okay? So as a result, the PDH, so pyruvate dehydrogenase kinase, is activated by NADH and acetyl CoA. So PDH kinase is what's going to phosphorylate here. So that makes sense. And these are kind of how we regulate PDH allosterically and covalently. And remember that this matters a lot because pyruvate can have about 1 million different options, right? Once again, it is the most connected metabolite. Within just the Tca cycle, it can make acetyl CoA, or it can make oxaloacetate. And that's not to think about acetate. That's not even thinking about the amino acids that it can make, or the whatever contribution it has to fatty acids biosynthesis, or the million other parts it's involved in, or gluconeogenesis, or any of that. So it's very important that anything that involves pyruvate be heavily regulated, okay? All right, so I'm gonna pause here before, this is something we've covered actually, okay. So I'm gonna pause here before we move into plants and bacteria. Does anyone have any questions? Okay, awesome. All right, so the Tca cycle, we talk about you put in 2 carbons and you get out 2 carbons as CO2, but because you're starting with oxaloacetate, a 4-carbon thing, you end with 4 carbons, okay? Acetate, so not acetyl CoA, but just acetate, it would be completely metabolized and all lost to CO2, and we'd have no carbon available for biosynthetic reactions, right? And that's why plants, bacteria, algae, don't use the Tca cycle. They use what's called the Glyoxylate cycle. And these are all very weird pronunciations for me, so I apologize that it takes me a moment. Okay, so the Glyoxylate cycle bypasses a few of the reactions in the carboxylic acid, in the Tca cycle. So reaction 1 is the same. Acetyl Coa plus oxaloacetate to give us citrate. Isocitrate is also the same. But at this point, normally we would go through a dehydrogenase reaction. The isocitrate dehydrogenase to alpha ketoglutarate. Plants and algae don't do this. The glyoxylate cycle does not do this. The glyoxylate cycle will either go to exactly what you would think, glyoxylate, and then into malate, or it'll go into succinate and then just continue from there. Okay, so it bypasses alpha ketoglutarate, and succinyl CoA. And I apologize for my handwriting. Okay. So why does it do this? Because those are the two reactions that release CO2 that we want to avoid. Okay. And here's a kind of a better description of how this looks. So this is the actual cycle, and we're going to ignore succinate for a moment because it can enter, but it's not really the main part of the cycle. Okay, so we're just going to ignore this for now. So we've used one acetyl CoA. We convert to isocitrate as normal. Then we go to glyoxylate, and we use the second acetyl CoA to convert to malate, and from malate to oxaloacetate. So we use two acetyl CoA, and what do we get out of this? Okay, so that's something that I think is really important to ask ourselves because, I mean, the two reactions we're bypassing are two reactions that give us NADH. So we're skipping the two kind of bigger reactions, and we're skipping the succinyl CoA to succinate reaction, which would give us an ATP, or a GTP that's converted to an ATP, I should say. So we're left with one FADH2 and one NADH. So this is not as pro-energy as as our body does, but because plants would run out of CO2, they have to go through this way. Okay, and did I miscount? I did miscount. I apologize. There's two NADH. Right, because I think is it? Yes, here we go. Okay, it's counting because succinate is going to come in and make its own. I apologize. I forgot about that step. Okay. So yes, so excess oxaloacetate is used for gluconeogenesis, so we can get a little bit more glucose out of it. Great. But this is, again, per 2 acetyl CoA, whereas for us, a single acetyl CoA gives us 3 NADH and 1 FADH2. So this is relatively inefficient energy-wise, but it's there because they have to conserve their carbon. Okay. All right. So the glyoxylate cycle will occur in glyoxysomes. Fairly straightforward, but it still uses those same TCA intermediate or TCA enzymes from the mitochondria. Okay, so that's a kind of confusing step. So this, I've seen maybe 1 or 2 questions on glyoxylate. It's not the most common cycle, but yes, this is a fairly content-heavy exam, and that's why I've been favoring the slides a little bit more than questions, because I think that it's important to go through the content really well. Okay. I think that the most important topics are knowing the TCA cycle itself, which I would suggest just draw it out 2 or 3 times to get used to it. Knowing the regulation of the TCA cycle, at least generally. The more specific, the better, but if it's too much, it's too much. And let me think, there's one topic in particular that I really emphasized, the anaplerotic reactions. Those are kind of the 3 biggest things that I would suggest. The glyoxylate cycle is somewhat important, but is not the most important thing, okay? like depending on your professor, you might get one question on it, you might get two, you won't get a lot either. I always have to prepare myself to pronounce them. Yeah. Okay. Sorry, before I forget, because I didn't talk about this bullet point. The glyoxylate cycle helps seedlings grow in the dark using acetyl CoA for fatty acid degradation. Okay? So plants, we know they love light, right? Photosynthesis, all that stuff is plants. So they don't just use plants though, or I'm sorry, plants don't just use light because they get the light to help them make glucose and then they can use that glucose. But here we're seeing they don't just get glucose from light. They also use the fatty acid degradation in the same way that we do. So if we think back to, gosh, let's scroll all the way back up. Right here, we talked about how you can get acetyl CoA from the beta oxidation of fatty acids. Well, guess what? Plants can too. And so plants can be really resourceful in the same way that we are by using that, that fatty acid degradation at night while they wouldn't normally be active. Okay, all right. So this is how the glyoxysome kind of works. And I want to put, there are kind of 4 main units of entry and exit between the two cycles, or between the two organelles. So I'm going to start from kind of going this way in the cycle. So the beginning would be succinate. Succinate can be converted, or I'm sorry, succinate is taken from the glyoxysome into the mitochondria to be converted into oxaloacetate, okay? Because the glyoxysome does not have the Tca cycle enzymes. So these 4 reactions, mitochondria. And then you're left with oxaloacetate that we need to get back over to the glyoxysome because we need oxaloacetate for the glyoxylate cycle. So rather than just go through oxaloacetate transfer, because we can't do that, and for the same reason we can't in our own body, right? What we do is we use oxaloacetate, which is 4 carbons, and glutamate. Does anyone remember how many carbons glutamate is? No, that's all right. It's one of the weird ones, to be honest. It's don't worry about it. It's 5 carbons. Okay, so this is 4 carbons here, 4 carbons. This is 5 carbons. Okay, and we get aspartate, which is 4 carbons, and alpha ketoglutarate, which is 5 carbon. So we're never changing our carbon number. It's just we're converting them into forms that can travel through the between the glyoxysome and the mitochondria. So glutamate we get from the glyoxysome. Aspartate and alpha ketoglutarate transfer back into the glyoxysome. Okay, and then this exact same reaction is undergone here as we just did over there, but backwards. So now rather than converting to aspartate and alpha ketoglutarate, we're going to convert these guys into glutamate and oxaloacetate. So if you notice, glutamate's entire purpose is to be transferred, made into aspartate and alpha ketoglutarate, have those transferred back, be made again into glutamate, and transfer again. It's just there to transport oxaloacetate. Okay, so same with aspartate and alpha ketoglutarate in this case. We don't really care about that alpha ketoglutarate for its being a part of the Tca cycle. We care about it because it can travel between the mitochondria and the glyoxysome, right? And then we get our oxaloacetate. Oxaloacetate goes through this glyoxylate cycle. And then malate here can be converted into oxaloacetate, or it can be converted into oxaloacetate outside the glyoxysome, like we have in our gluconeogenesis, and then go through go into carbohydrate, just like in gluconeogenesis. Okay, and just fatty acids can also make acetyl CoA. So that's kind of the overview of the glyoxylate cycle. So there's a little bit more to this chapter, but that's really the bulk of these cycles. So before I get into this kind of extra stuff, does anyone have any questions about this? About anything that we covered today, by the way, not just the glyoxylate cycle specifically. Okay, all right, terrific. So I completely understand. So Pep Carboxy Kinase. This is one of the gluconeogenesis intermediates, and specifically, this is the one that converts oxaloacetate into Pep. Okay, so it is overexpressed in skeletal muscle. So let's think about why. So mice, their Pep Carboxy Kinase is 7 times more active, and they have enhanced exercise capacity. So, in other words, mice are able to, rather than go through lactate generation, they use fat degradation extensively, and they have a lot more mitochondria and a longer lifespan because of that. So, what does that mean? It means that Pep Carboxy Kinase, or really just generally speaking, gluconeogenesis is a reaction that mice are able to do, but they're not just getting the gluconeogenesis, they're not converting glucose to pyruvate and then back to glucose. That wouldn't make much sense. They're converting their fatty acids into pyruvate, or into acetyl CoA rather, acetyl CoA into pyruvate. Well, yeah, acetyl CoA into pyruvate, then going through gluconeogenesis, and then using that glycolysis to get really quick energy. So because of that, they're able to use their body fat extensively to give them energy. Okay, and there's a picture. Okay, so yeah, so that's kind of what we've been talking about here. So mice violate one of the pillars of aging research, namely that limiting food intake increases longevity. It's completely fascinating, but basically, I'm going to see if I can try to keep this organized because there's a lot going on. So mice have glucose. They convert the glucose to pyruvate, to oxaloacetate, into the Tca cycle. The proteins and the fats, usually mostly fats, that mice also consume, they convert into oxaloacetate to be converted by Pep Carboxy Kinase into Pep for gluconeogenesis, so that they can get more energy out of it. Okay, and once again, it comes down to that idea: aerobic versus anaerobic respiration. If they can go through the Tca cycle, they will. If not, gluconeogenesis is needed so that they can get some energy. Okay, so this is kind of just an interesting thing your professors like to include. I don't think it's ever been tested on. It's just a fun fact. Okay, so before we conclude for the day, I just want to very briefly kind of go over the most important topics. So the first, like I said, the actual cycle. The second is the regulation of the cycle. The third is, how did I already forget the third? Is the anaplerotic reactions. There we go. And the fourth is this right here. The pyruvate dehydrogenase complex with the 3 enzymes and their reactions is one of the things your professors really like to test on, because it really tests, did you learn it or did you just memorize a couple of the key terms? Okay. So that's really all I have planned. Does anyone have any questions, or should I stop the recording? Okay, awesome.